two keynote speakers, Dr. Rafi Ahmed and Dr. Vijay Kuchroo. And the session is being moderated by Professor N.K. Mehra. You all know Professor N.K. Mehra, but I'll introduce once more. Professor Mehra, you all know, was former Dean of All India Institute of Medical Sciences. He is ICMR Emeritus Scientist. Earlier, he has served as a national chair for 5 years and professor and head of Department of Transplant Immunology and Immunogenetics at All India Institute of Medical Sciences. He's the one founder member of FIMSA also, and he was Secretary General of FIMSA. Besides all his other achievements, he has been able to conduct many FIMSA programs. He started with that, and then a couple of them we conducted in Chandigarh also. Last year, he conducted a very nice meeting in Jaipur. So I just invite him to moderate this session. Professor Mehra, please take over.
Thank you very much, Sunil. We are really in for a very wonderful session. This is going to be the last session of this course. And as they normally say, that we keep our best towards the end. And we're going to have two eminent speakers from around the world. Number one is going to be Rafi Ahmed. Rafi is a very popular name in India, but here there's a different Rafi we are talking about. The science Rafi there. Rafi Ahmed is, of course, the Georgia Research Alliance Professor of Microbiology and Immunology and Director of the Emory Vaccine Center at the Emory University School of Medicine in Atlanta. His research efforts are directed towards understanding the mechanism of immunological memory and using the knowledge to develop new and more effective vaccines. I can tell you, I haven't ever heard a better lecture on immunological memory than from him. I am reminded of that year, I think about 8 or 10 years ago, when he came to Goa. We organized, Vijay and others had organized a course over there, and he had brought several slides, but he could not go beyond 5 slides because, within the 5 slides, the generation of discussion was so high. But this is not going to be the case over here. I have already noticed that he has 40 slides to show you, but it's going to be a feast for you to understand the immunological memory. The number 2 issue that his lab works is on defining the mechanism of T-cell exhaustion during chronic viral infections. And you saw it yesterday that we were talking at least in COVID-19, there is so much of T-cell exhaustion. And T-cell exhaustion is known to be in chronic viral infections, in cancer, and his lab is developing strategies for restoring functions in exhausted T cells. Dr. Rafi Ahmed is a member of the National Academy of Sciences and a member of the National Academy of Medicine. I really don't want to be between you guys and Rafi. Rafi, the floor is yours. You're going to speak for about 45 minutes, and then we will have a number of questions from the audience.
Okay, thank you, Narendra. Very kind of you. Yes, I do remember the Goa meeting. It was very interesting that that you and Abdul Abbas and Vijay had it. Yes, and I wish again that we were doing this in person. One of the nicest things about being part of conferences in India is that I get to come back to my home country. but this time we are living in the world of Zoom, and so this is how I guess it will be for at least for the immediate future. I want to thank Amit and Sunil for the invitation and really organizing this wonderful course. And again, it's a pleasure. I kind of changed the title of my talk, and I'll tell you why I did that, because when I looked at the program, there were several excellent talks earlier on there was very nice talk on at least on the program, by Joshi Jacob on B-cell memory. There were another nice talks on tissue resident memory cells, and a lot of talks on memory T-cell differentiation by other people. And what was kind of missing, although I think Vijay will also touch upon that same theme, what really was missing was what happens during chronic viral infections to T cell differentiation. So that is the topic of my presentation. And let me see if I can go back to my screen sharing and my talk. Okay, so let's see if I can.
There you are. Good.
This navigating Zoom is the hardest part of this giving the talk. Okay, all right, great.
Yeah, there we are.
All right, let me go back. Okay, so the title of my talk is T-cell lifestyle in chronic viral infection. And in my talk today, I will cover two topics. One, in the first two-thirds of my talk, I will kind of summarize work that we have done and also others in the field, which define the CD8 T-cell states during chronic infection. And one of the points that I will make throughout the talk is that the differentiation program of cytotoxic CD8 T cells is very different during a acute infection that is eventually cleared, whereas a chronic infection that's going to persist, especially with high levels of antigen for many months or many years. And what I'm showing you today, these paradigms also hold in cancer, in human cancer, and also in many human chronic viral infections. I will then, after defining or summarizing these CD8 T cell differentiation pathways and the different subsets that exist, I will then end my talk on a few slides about the implications for immunotherapy based on the knowledge that we have about CD8 T cell differentiation. So this cartoon slide summarizes work that we and many in the field have done over the last, I would say, 25 or 30 years. And I'll just walk you through the simple cartoon. Uh, when, uh, when a naive CD8 T cell encounters antigen, it undergoes a program of very rapid proliferation that is coupled to differentiation. And this results in a large number of highly cytotoxic effector CD8 T cells, which are generated. These effector cytotoxic CD8 T cells play a major role in controlling the infection. These are the cells that will go to wherever the virus is, kill those infected cells, and result in very efficient viral control. And especially during a primary infection, these cells actually become active very early, even before one has much neutralizing antibody. This effector CD8 T cell response starts eliminating and killing the virally infected cells. Of course, antibody is also very important in eventual eradication or control of the infection, and becomes very important later on in terms of reinfection. But in controlling a primary viral infection where there really isn't any pre-existing antibody, the effector CD8 T cell response is a major player in eliminating and controlling the infection. After the infection is cleared, these cells will now further differentiate and give rise to functional memory T cells. This is an oversimplification of functional memory, but essentially the bottom line here is you will have memory CD8 T cells circulating in blood. You will also have memory CD8 T cells in tissue, referred to as tissue resident memory cells. These cells, upon re-encountering the virus again, seeing it for a second time, can elaborate effector function very quickly, can proliferate and control the infection. So this is the good scenario that happens after most acute viral infections. Good effector CD8 T cell response, very large number of these cells are generated. Majority of these effector cells will undergo apoptosis and will die, but a subset survive to give you a pool of long-lived and functional memory CD8 T cells. This is the good scenario. But when the virus persists, especially if it persists at very extended high levels, then these cells become dysfunctional. Term that we used many years ago was exhaustion, when you describe that actually there's less function in these cells. And work that was done by Dan Barber, who was a student in my lab, showed that the major regulator of this T cell exhaustion was the inhibitory receptor program cell death 1, This is a molecule that was cloned and identified by Tasuku Honjo from Japan, and he was recognized by the Nobel Committee a few years back for his discovery of this important molecule. What the studies of Barber showed is that PD-1 is the critical regulator of T-cell exhaustion, and blockade of this pathway results in more increased function of these cells and reduction of the viral load. These original findings were made by us using the chronic LCMV infection model, and many others then extended these findings to human chronic viral infections like HIV, hepatitis B, hepatitis C, and so on. And then the people working in the cancer field made very similar observations. Today, what I will do is now jump forward many years from these original findings, and then actually jump forward 10 years, I would say, and then give you our current understanding of what we really mean by T-cell exhaustion. What are the different types of CD8 T cells that are present under these conditions? And then how is this response being maintained in these chronically infected mice or humans for that matter? All the studies I'll present to you today come from this mouse model of chronic LCMV infection. Basically, we're just taking it in this scenario. So a major breakthrough in understanding the different T cell subsets and how this program is maintained was described 4 years back by Seijin Im, who was a postdoc in my lab. And this paper, Seijin described this and identified the cells actually that proliferate upon PD-1 blockade. And then he showed that these cells, which I'm going to go into some detail and describe to you, are also the cells that maintain this ongoing T-cell response under chronic conditions. Even though this is not very successful in totally eliminating the infection, it does keep the virus in check. And there's some control, but not complete control. It's like a détente situation between the immune response and the viral infection. So I'll spend quite a bit of time describing to you what this cell looks like. And then others have also made similar observations in the mouse LCMV model and also in other studies. A second paper that was published by Will Hudson, who's currently a postdoc in the lab, defined the transitory population that arises from the stem-like population during this chronic viral infection. And again, others have also made similar observations. So the next few slides I'll show you are really cartoon slides, which will summarize the findings of these two studies. This is a busy slide, but I will walk you through it. Okay. So the key finding of Seijin was identifying what I will refer to as either the resource cell or the stem-like CD8 T cells. And everything I'm going to show you is looking at antigen-specific cells using the MHC class I tetramers. So we know that when we are describing a cell population, it indeed is specific for the virus, okay, based on the MHC class I tetramer staining and then the detailed characterization of these cell populations. So what we found that was really interesting about these cells is that even though these cells are persisting and are present under conditions of a very heavily infected animal or heavily infected condition, these cells actually are very quiescent. They're very quiescent cells, and their location is very interesting. They're found really only in lymphoid tissues. This is a viral model where there is virus in every tissue in the mouse, both lymphoid and non-lymphoid. But these cells are only in lymphoid tissues. And later on, I'll quickly show you a data slide that not only are they in the lymphoid system, that is in the spleen, the lymph nodes, a little bit in the bone marrow, okay, but within the spleen and the lymph node, they are actually resident within the T cell areas of the spleen, i.e., these cells are not circulating in the blood. They are resident cells and they're in the lymphoid compartment in the T cell zones. So what characterizes the properties of these cells? First of all, they actually have significant proliferative potential. So they are very quiescent. They can undergo a slow self-renewal, and they have proliferative capacity, which is being regulated by PD-1, and I'll get to that in a second. If you look at their expression markers, they express PD-1. The key transcription factor they express is TCF1. They also express BCL6. TCF1 is essential for the generation of these cells. They express high levels of costimulatory molecules. Even though they express PD-1, many of the other inhibitory receptors like TIM3, CTLA4, 2B4, many of these others well-defined are not expressed by these cells. They have no effector molecules. They're very quiescent cells. They express interesting set of chemokines and chemokine receptors. They respond to CXCL13. That means they could go to T and B cell borders because they express CXCR5. Importantly, they respond to CCL19 and CCL21 because they have low levels of CCR7 that allows them to go to these. So these are all things that's going to keep them within the lymphoid system, within T and B cell borders. And in cancer, these cells actually are found very often in the tertiary lymphoid structures because again, they respond to these chemokines which are present in tertiary lymphoid structures. They express XCL1. This is a very interesting chemokine that will attract the cross-presenting DCs, the XCR1 DC. So they're sitting in a sweet spot of the lymphoid system surrounded by good DCs. It's this cell that is at a slow rate continuously generating a transitory population. When this differentiation happens, the first thing that happens is downregulation of TCF1, upregulation of TIM3, the cells start proliferating, they express many effector molecules. This transitory population is actually reasonably functional. These cells come out in the blood, then they go to multiple tissues, wherever the virus is. Now they further differentiate there, and then over time become very dysfunctional. This is the one we refer to as the exhausted cell. So this is a small population residing in lymphoid tissues. These cells come out in the blood, and then these are the largest number of cells in many lymphoid tissues, which express very high levels of inhibitory receptors. They do have many effector molecules, but this function is constrained by the environment and also the multiple inhibitory receptors that they have, that they express. Then they express very few co-stimulatory molecules. So what happens when you do PD-1 blockade? When you do PD-1 blockade, this whole thing now happens much more faster. If this was happening at rate 1x, after the blockade now, it happens at rate 10x. That is, many more of these cells now differentiate, and that what that tells you is that the PD-1 on these cells is keeping them quiescent, keeping them in the sweet spot there. And but once you remove that break, one of the few inhibitory breaks which is on these cells, you now get very rapid proliferation and differentiation of these cells. And then these cells come out in the blood, go into the tissues, and there, because the PD-1 will block interaction at the effector phase, can now elaborate some viral control. So let me first address quickly, showing some data. I'm going to go through the data part very quickly because I think some of this is published, some is not, but I don't want to spend too much time, but give you the bottom line message. So one of the first questions we asked was, in these chronically infected mice, do you need this continuous input of these, do these cells need to continuously be generated from naive cells? And also the other question we asked even before is, when do you get the generation of these two subsets? And the answer is, it actually happens very early. And this is a very important point. So when do you get this bifurcation of these terminally differentiated cells and the stem-like cells? Quite early, during the first week to 10 days of this chronic infection. Of course, all the response initially comes from the naive cells that proliferate during this period, but there's a program, and TCF1 plays a major role in it. The cells that re-express TCF1 become the stem-like CD8 T cells. These become these terminally differentiated cells. These have a very short half-life. So within a few weeks, these are all dead. Okay. This cell, of course, survives, and this is the one that keeps the engine going. So we asked the question, if you don't have any more naive cells coming into the system, can this maintain the response or not? So and that's the question we asked, whether you need continuous input from the thymus to maintain it, or if you have sufficient numbers generated initially, can that keep the system going? So this was done by using thymectomized mice. So the experiment now would be using thymectomized mice so that adult thymectomized mice, so they can't continuously be generating these cells. But if once this is generated, can you keep the system going? And the answer is yes. And I'll quickly go through the data. Okay. So in this case, what we are comparing are responses in either thymectomized mice or the sham thymectomized mice. Again, thymectomy is done in an adult mouse, so it already has many naive cells in the periphery. That's what will generate the response. But later on, there is no thymus there, so you cannot maintain that input long term. So what you see here is whether it's thymectomized or sham thymectomized, the number of antigen-specific cells are very similar in these two individuals. And if I break it down and ask how many of the terminally differentiated cells there are, they're the similar number, and how many of the stem-like cells there are in lymphoid tissues, they're the same number. This is a second epitope from LCMV, same pattern. I'll quickly show you one data slide. It's a busy slide, but just focus on the things that I'm going to highlight. So this is at day 8. On this axis is the MHC class I tetramer. There's PD1 on this axis. Here are 4 mice which were thymectomized. Of course, they started out with a large number of naive cells at that time. They generated good response. Similar response is seen in the sham thymectomized mice. And here are the 2 subsets. These are the terminally differentiated cells. These are your stem-like cells expressing TCF1 and negative for TIM3. So you can see that both subsets are generated at this as early as day 8. And these are going to be the important cells for generating. These are the more terminally. At this time, they've all been generated, of course, from naive cells initially. Now we look at a later time point, and we have looked now actually as late as 6 months or even a year, and the pattern holds the same. So you see now that both the tetramer positions are maintained, but here's the important part. You again maintain at this time point, thymectomized mice have both subsets, and the sham mice have both subsets. That is, thymectomy did not impact it. That is, even in the absence of new cells coming, they are there. So this makes a very critical point, which is once you've generated these cells, this is the engine. These are the cells which are keeping this response going. So a prediction of this is that if you look at the T-cell receptors of these antigen-specific cells, there should be similarity between these. And the answer is yes. We published a paper recently in Journal of Virology. Danny Chang, who's a PhD, MD PhD student, showed this. And what this showed is that almost all of the TCRs present here are over here. Remarkable overlap. That is, this is what keeps the response going. Okay, then the next question we asked was, if you do single cell analysis of these different cell populations, that is, you sort with the tetramer, do single cell RNA-seq, and see what are the different genes which are expressed by these cell populations. And here is an example of that. You see these 3 clusters coming up: the stem-like cells, the transitory cells, and the terminally differentiated cells. These are your naive cells. And I'll just point out a few genes which are expressed by these 3 different subsets, which I've mentioned to you earlier, but now I'll show it to you at the single cell level. So as I mentioned to you, PD1 is expressed by all 3. The stem-like cells express it, transitory cells express it, and the more exhausted cells express it. Of course, naive cells don't express any PD1. This is a transcription factor, which several groups have shown is important for this program during chronic infection and cancer. Again, all of these cells express STOX. TCF1 only and exclusively expressed by the stem-like population. So here you've got and naive cells also express TCF1. TCF1 is very important transcription factor for good T cells. Naive cells express it. Memory cells after acute infection express it. But in this chronic thing, it's only this population, which I'm referring to as the stem-like or resource cell, that expresses TCF1. But note that this cell is also expressing TOX, which a naive or a memory cell will not. And it also expresses PD1, which a naive and memory cell will not. So a very unique expression program of these cells. They don't have TIM3, it only comes up later. This is another exhaustion marker, which comes up later, is not here. These cells, the stem-like cells, have no perforin, have no granzyme, because again, they are quiescent cells with this resource potential. Okay. Granzyme B, perforin comes up as they differentiate. They express high levels of co-stimulatory molecule. This is very important in terms of rescue from PD-1 blockade. Again, they express these. IL-7 receptor needed for long-term survival, signaling through CD120 needed for signaling through IL-7, which is critical, but these cells are long-lived. Okay. They express IL-7. Common with naive cells, okay, but again, very distinct from naive cells because they express PD-1, they express TOX, and so on. And XCL1, again, uniquely expressed by these cells, so that in this niche they're sitting, they recruit and attract good DCs around them. Again, I want to emphasize that this population that we and others have defined during chronic infection is very unique cell. And these chronic stem-like cells or resource cells are very different in their gene expression program and also in their epigenetic program from effector cells that you will get during acute infection or the TEM or the TCM or the TSCM. Some genes and properties are shared, but this is a very unique cell population you find under conditions of chronic infection. I'll get back to this more in a few slides. So what are the migratory patterns of CD8 T cells during chronic infection? And as I've shown to you earlier, really the stem-like cells are resident in lymphoid tissues, and the differentiated cells actually are resident in the non-lymphoid tissues. So we did this this paper also was published earlier this year by Sajan. This is a complicated slide, but the experiment here was doing parabiosis between two chronically infected mice. As a control, we have here immune mice, which have cleared the infection. These will equilibrate, that is, cells from here will go here, cells from here will go here, because these memory CD8 T cells are circulating in the blood, what we refer to as the TCM. These will nicely equilibrate. But if you do the parabiosis between two chronically infected mice, there's hardly any migration. Basically, the cells of this mouse stay here, the cells of this mouse stay here, just a little bit of cells come out. Okay? And the cells that come out a little bit are those transitory cells. So basically, this is what you have. You've got the stem-like cells, which are tissue resident in lymphoid tissues. They express high levels of CD69, which keeps them in these, a canonical marker of resident cells is CD69. They express high levels of CD69. They express low levels of S1PR1, which is an exit molecule for cells coming out, and also low levels of S1PR5. When this first transition differentiation happens, CD69 goes down, S1PR1 comes up, but also S1PR5 comes up, which is a exit receptor that comes up on recently generated effector cells. So you can see this transit population in the blood. And then once they go to the non-lymphoid tissues, again, CD69 goes up and these two come down. So the situation you have is small number of these cells residing in lymphoid tissues, resident, very large number of these in the tissue, which are also resident there. And then the circulating population, transitory, which comes up, which is in the blood. There is very few cells are in the blood during chronic infection. Okay. Of course, this increases greatly upon PD-1 blockade. All right, so let me conclude this part of the talk, where I think the major message I want to give you is the identification of this unique cell population. And we feel that this is an essential adaptation of a T-cell response under conditions of chronic antigen stimulation. Let me take a step back. So if once you have a differentiated CD8 T-cell, its half-life actually is not very long, especially if this is a differentiated CD8 T-cell, which is continuously engaging many non-lymphoid cells, which don't have appropriate co-stimulatory molecules, and you're continuously engaging the TCR of a differentiated cell. This is a recipe for the death of the effector cell. Most effector cells have finite lifespan, and in our experiments, the more differentiated cells only live for about 2 to 4 weeks in these mice, maybe a little bit more, but not long term. So the only way to keep the response ongoing is to have a cell that is continuously generating these responses. And this is the unique cell that is hiding in a very sweet spot within the lymphoid system and slowly generating an ongoing response. And again, as I said, when you remove the PD-1 break, this is the one that comes out and gives you many more of these newly generated. So with this, so we feel that this resource cell is essential for maintaining T-cell immunity under conditions of chronic infection. And we would propose that these cells would be not only in chronic infections, but also in cancer and in autoimmunity. And indeed, many papers have been published by others looking not only in the viral mouse models, but also in HCV and HBV and HIV, finding similar cells in humans as well. And I just show you a few papers here from in human cancer that are now probably 20 or 30 or even more papers in human cancer, which have made very similar observations and defined this unique cell population. And also some papers are coming out in autoimmunity, which are finding very similar cells. I think, and Vijay will hopefully address and expand on some of these. Again, this is an earlier thing and many more have come out. So again, this is the main message I want to leave you with for this part of the talk, is the identification of this unique population, which is critical for maintaining an ongoing CD8 T-cell response. And that's why the title of my talk was CD8 T-cell lifestyles in chronic infection, because this has to adapt to living with antigen, but finding a sweet spot where it's hiding in the right environment. And also these cells have a long half-life. They basically can live for the life of the mouse. They can also undergo a very slow self-renewal. So let me quickly shift to and make a few comments, and I think I have about 10 minutes left or so, or 15 minutes, and I'll quickly go through this thing about implication immunotherapy. There's fair amount of data here, data slides, but I'll go through that quickly and just emphasize the bottom line message. This kind of just summarizes our view of how PD-1 blockade works. And so essentially you have these stem-like cells sitting in the T-cell zone, and they are being regulated. PD-1 is still restraining them. So when you do PD-1 blockade, the first thing that happens is removal of this break results in the proliferation, differentiation of these cells. Note that this is really the main cell that expands after PD-1 blockade. PD-1 blockade resulting expansion has been documented by us and by many others in humans. You get this big burst of cells come out after the first infusion of PD-1, and that is coming almost exclusively from the cell population. And then these cells go out into the infected tissues. In the case of cancer, would go to the different sites of where the tumor is. And then PD-1 blockade is also working at the site of the target. It is a very key thing which people have ignored. It's not just the proliferation. You also have to block this negative interaction at the site of infection or at the site of the tumor. And this is what results now in these, what we are referring to as the terminally differentiated cells sitting in the tumor bed or in the virally infected tissues, which are being prevented from killing the target cell because of this negative interaction. This blockade results now in release of granzymes, perforins, and many different cytokines, which result in reduction of the viral load or the tumor in the case of tumors. This expansion is very dependent on CD28. So a paper that was published by Alice Camphorst, a previous postdoc in the lab, showed that if you do PD-1 blockade alone, it's not sufficient. They also need to get these co-stimulatory signals. So if you do PD-1 blockade and block CD28 B7 co-stimulation, you don't get this expansion. So not only is it essential to remove the break, but also to provide co-stimulatory signals. And again, I go back to where they're sitting and what they express. Stem-like cells express CD28. These terminally differentiated actually don't have CD28. And these are surrounded by good DCs so that they can get co-stimulatory signals. Again, this was the paper that showed this CD28 dependence of expansion after PD-1 targeted therapy. So let me end by a few slides about combination therapy. So one of the major emphasis now is doing combination therapy. PD-1 blockade by itself is effective to a certain degree in multiple cancers. It's been licensed for use in all of these plus more. But response rates, still many patients don't respond, or the response is only for a short period of time. So there's now a major emphasis on combination therapy, and almost many different approaches are being used by many labs, many pharmaceutical companies. And I just want to uh, end my talk on a few slides about cytokines, that is combination therapy with PD-1 in combination with cytokines. And in particular, I want to just share with you data on IL-2. Basically, we looked at the gamma chains. We looked at comparing PD-1 blockade with IL-2 or IL-7 or IL-15. All of them are beneficial in different ways, but I just want to focus on what we see with IL-2. And the reason I'm focusing on this is because, at least in our studies, we have found that one of the best combination therapies is actually PD-1 with IL-2. It's very simple. One of the more simpler things actually has been, at least in the model that we have looked at, highly effective. And our original paper was published in 2013 by Erin West, who was a graduate student in the lab, and she showed very good synergy. But what I want to get, but this was done before we identified or discovered these stem-like cells. So we now are going back, and what I'm showing you in the next few slides is all unpublished data. We are now going back and revisiting this and asking, when you do this combination therapy, what cell is IL-2 acting on, and what is happening at a cellular level now that we have a better understanding of the different states that are there, cellular states that are there in the chronically infected mice. So this is a question: which subset responds to the combination therapy? So the experimental design here, this was what Sajan had done in his Nature paper. You have a chronically infected mouse, you isolate the stem-like population, and you isolate the more exhausted cells. You adaptively transfer these cells back into a infection-matched mouse, okay? And you use different congenic mice so you can identify the donor cells from the recipient cells. And this is the published data saying that when you do this experiment and you do PD-1 blockade, almost all of the response comes from the stem-like cell. So this is after transfer stem-like cells, untreated mice, small number, PD-1 blockade, significant expansion and differentiation. And if you transfer these more terminally differentiated cells, basically they have no proliferative capacity. Certainly after blockade, they can kill cells, effector function, but zero proliferative capacity in this population. So we did a similar experiment, or I should say Masao Hashimoto, postdoc in the lab, was working on this project, did a similar experiment and asked what happens after IL-2 thing. That is exactly the same design as before. Okay. And then you treat, now you do combination therapy, and also had a group with IL-2, but I'm just going to show you the combination therapy. The question is, when you combination therapy, are you also getting a response from here, or is it still these cells only? And the answer is it's still these cells only. So they respond not only to IL-2, but they also respond to PD-1 blockade and the combination. And here's the number, really striking increase in numbers. So if you transfer the stem-like or the resource population coming from a chronically infected mouse back into a congenically infection-matched mouse, do the untreated, no treatment, very few of these. If you now do combination, huge increases over 3 log. PD-1 alone would give you something over here, now much greater, about 10-fold greater than what PD-1 alone will give you. Of course, this is coupled to differentiation. I'll get to that. So huge numbers in the spleen, liver, lung, everywhere. And the viral reduction effect is also the most striking in these in this combination therapy. Then we asked what turned out to be the most interesting question that interesting results that we got. So I've shown you in the previous one that after combination therapy, the proliferative burst is coming again almost exclusively from that resource population. But then the question was, what about qualitatively, what do you get? As I've shown you before, is the resource cell that gives you the transitory cell and then the more differentiated population. Okay. And after PD-1 blockade, you get the same thing, and those cells qualitatively look very similar. But the question we had, what about after the combination therapy, where the cell is not only getting nice IL-2 signal, not only has the PD-1 break removed, but is also getting nice co-stimulatory, nice IL-2 signals? What will happen under these conditions? And the results are that they are strikingly different. And that's shown over here. This is RNA-seq analysis. In this case, and everything again is done on the MHC class 1 tetramers. This is comparing the untreated versus PD-1 blockade alone. They're very similar. You have many more of them, and PD-1 is there doing its good thing. But transcriptionally, the cells are very similar. That is, you have more of the same, which is very good, but qualitatively, they're not different. But in this combination therapy, you find striking differences. Many genes are upregulated, many genes are downregulated in these new effectors which are being generated by combination therapy. The next few slides are single cell RNA-seq showing you how strikingly different this population is. So here is, let me walk you through this one. This is single cell RNA-seq of 3 populations. There either we have tetramer positive cells from the untreated mouse, which is in yellow. So here are your 3 populations, the transitory, and then the more exhausted and the stem-like cell. Okay, stem-like, and these 3 are over here. And then the PD-1 blockade, you stay pretty much in the same place. Here are naive cells. This is the combination. Basically, you get a totally distinct and unique cluster. So the product that's coming when you give both of these signals is generating now, is now a very, very different cell. And here are some examples of what happens. So in this case, you find that this is the, let me just go back here. This is the cluster you want to put your focus on, the one on the left-hand side. This cluster is only generated in the combination therapy. And you can see that this actually has lower levels of PD-1 than these, lower levels of tox, that is the inhibitory signature is decreasing. And many of these cells also have TCF1. This is the stem-like cell. In this more differentiated cells also have these good molecule there. They have KLF2, which is an exit receptor. They express high levels of S1PR1, very high CD127, very high IL-18, okay, and very low levels of TIM3. So the effector cells you get now basically have more of a, after combination therapy, more really have a good effector signature. And I'll elaborate on this very quickly by also looking at the epigenetics. So this is just a small flavor of a few genes, and I'll summarize this in the next few slides. But what about epigenetically, what's happening? And again, here, striking differences. This is by ATAC-seq of antigen-specific cells comparing the untreated versus the PD-1 blockade. Very similar, some minor differences, but look at this, massive differences. Upregulation of many, opening of many genes open up at the epigenetic level, many actually get closed. And the ones that open up are again all related to effector function, migration, and so on. And this is now a summary slide, which is showing you how different, first of all, cells are during chronic versus acute, and then how the combination therapy starts moving it in the good direction. So this is how the ATAC-seq of naive cells, very different. These are the good effector cells and memory cells you get after an acute infection. This is where cells sit epigenetically in terms of their program after chronic infection, the stem-like terminally differentiated, and after the PD-1 blockade, they still remain very similar epigenetically and transcriptionally. But now in the combination therapy, they're becoming much more effector-like that you see during an acute infection. So this is, I end on this saying that what this simple combination therapy is giving you is the product here are cells which have increased effector signature and decreased exhaustion signature. And that is the reason why you get this much better control of the infection. So let me end here by saying that as we go forward in designing combination therapies, we should keep this in mind, this pathway. And which cell to target, as we've shown, the IL-2 is targeting both of these and giving us much better effector population, which is resulting in much more efficient viral control. So let me end here by acknowledging the key people who did this work. Sejan M really did the breakthrough work identifying the cell population. Will Hudson defined its transitory population that I mentioned to you. And then Masao has done all the work, this unpublished work on this combination therapy. Many wonderful collaborators over the years. Thank you very much, and I'm happy to take any questions.
Thank you. Thank you so much, Rafi, for that masterly talk. Fantastic. We always enjoy hearing from you. Now, there are a few questions I can see in the question box, but I have a couple of them, very simple ones.
Sure.
I want you to
Yes.
know, of course, in number one, you show this three transitional slide. I mean, the cells, they stem like the resource cells, they go through the transitionally phase, and then they are the terminal differentiated cells. This is in mouse. Is there any evidence also of this similar kind of situation in humans?
Yes, actually the best data, yes, there is data in humans, but the best in chronic infection, there are a few papers that have been published with hepatitis B and with HIV, and Rama Amara at the MD Vaccine Center has made similar observations in the SIV non-human primate model. But actually the best data is coming now from cancer. So there are probably 100 papers published already, almost every month. Some very nice papers come out. So these cells have been identified in lung cancer, in melanoma, and we actually have a paper that's currently under review in HPV, in head and neck cancer, where we have actually used, so most of the papers that have come out, the beautiful papers that have come out, have been bulk populations coming from the TILs. And not really looking at antigen-specific cells. We have some unpublished data that's currently under review, looking in HPV, human papillomavirus, HPV positive head and neck cancer. So here we actually used HPV specific cells. We looked at those in head and neck cancer patients in the TILs, and amazingly, we find exactly these three populations within the cancer. And you find the stem-like cells or resource cells are located exclusively in the tertiary lymphoid structures. So they are not in the tumor bed. They are in the tertiary lymphoid structures, but the more differentiated populations are in the tumor bed itself. So tumor stroma contains the transitory and the terminally differentiated population, and the tertiary lymphoid structures and the lymph nodes contain the stem-like cells. And we are very confident of this because we've used the MHC class I tetramers to identify these populations. So the short answer is yes, human cancer, very good documentation. A few nice papers coming out on autoimmunity are also showing something similar.
It might also be interesting to see them in COVID-19 as well, because COVID-19.
COVID-19, yes, you might find them during this early stage, but whether you get the same program or not remains to be determined. But again, I think anything that's persisting for extended periods, you might have something, yes.
I didn't understand your, the CD69 role. You said CD69 is increased in the resource cells, then they come down, and then they again go up over there. Why is this transition or a temporary sort of going down?
Yeah, because yeah, that's a very good question, actually. So the program they have in their original state when they're sitting in the lymphoid tissues is a program of being resident. So when the cells are resident, they express CD69, because that is work done by Jason Sister many years back, showed that CD69 is a marker for cells which will not be circulating. Also, S1P1R is low in those cells. So very nice interactions were defined as CD69 being a marker for tissue resident cells. So that's why. But once the transcriptional program changes, when they differentiate, they have to come out in the blood. Otherwise, they're useless. So they have to go in the blood. So then CD69 goes down, S1PR1 comes up, S1PR5 also comes up. Okay? Yeah. And then when they're back in the non-lymphoid tissues, CD69 again comes up. Okay? And again, because they then become resident in the non-lymphoid. So that's work. So those markers very nicely match their migratory properties.
Is it possible to say that the terminally differentiated CD8 positive T cells are now sort of end cells and there's no memory in them?
Yes, they are end cells. We have not found anything that reverses their phenotype or their transcriptional program. There might be, but we have so far not succeeded in doing anything to them to make them proliferate or to go back or to de-differentiate. There might be mechanisms, but the simple things we and others have tried don't happen. But they are important because I think those are, cells are eventually, when you have all the right blockades done, they are certainly contributing to the killing of the tumor cell or the virally infected. But they're the best effectors are the newly generated ones.
Okay. Good. Now I must go to the question box. I have to act like a good moderator. There is a question for you that in chronic infection model, you saw that the resource cells are generated as early as 8 days. So what
Yeah.
happens in the acute infection? Do they
Yeah.
also
Yeah, that's a very good question. And we are all trying to understand. Yes, that is, I think, one of the more interesting. And at this stage, why? So I think it's the environment. It's the environment. It probably has to deal with what cells are presenting antigen, but also, I think, very importantly, the microenvironment during the chronic infection, the different cytokines that are being made. I think those are influencing. We have some preliminary data that certain cytokines might be responsible for it, but it's too early to even mention it because it probably will turn out to be wrong. Okay. But I think that's a very key question. This fate decision happens early, uh, and what gives us this unique stem-like population, which you don't see in the chronic. In the chronic, in the acute, you have a similar program, but there the pathway that differentiates it is the more cells that will become long-lived memory cells versus the more term. So there are similarities, but the transcriptional uh, program and epigenetics of the stem-like cells are very different from what we have defined as the memory precursor cells during acute infection. So very early, both bifurcations happening, and all the cues are not fully understood. But that's, I think, a major question for all of us to address in the future.
That question was from SS Prakash. Now, Arendra Arya, a very good question. What is the role of viral load in the generation of CD8 stem-like T cells?
Yes, a major role. So a lot of what dictates the generation of these cells is indeed the viral load. And that's what I was getting at, that the higher the antigen load, the more likelier to generate. In some ways, the immune system is sensing that this is a virus that could persist for long term. And that's what triggers it. You have to act very early to give you those cells. The immune system in that way is beautiful. It has adapted to deal with the infection that is controlled, and you get the good long-lived memory cells you have after antigen is cleared. But if the immune system senses that this infection may not be regulated, very controlled very quickly, because it's such a high load and other cytokine milieus are changing, then it generates this cell. So it's a very early sensing to generate this population very early on, to be prepared to have this cell. Because I think if this differentiation doesn't happen very early, it may not happen. So that's why this sensing is happening early stage and is almost very heavily dependent on the antigen load. If the antigen load is low at that time creating, you will not generate this population.
I mean, you're going to find them more in the hospitalized.
Yes, you might have them more, exactly. So I think that, yes, I
Where
think,
innate
yeah,
immunity is sustained and the adaptive has gone down.
yes.
Now, there is another question from Sirijit Roy. Can blocking the PD-1 would increase the immunological memory in all kinds of viral infections?
Typically not, because the PD-1 is a major regulator really of under conditions of chronic infection. And there it's coming up for a very important reason. It's coming up to prevent excessive immunopathology. Because during that early phase, the CD8 response is very vigorous. When you have a heavy infection, many tissues involved, a very large number of effector cells are generated. And if in this early stage where there's a lot of effector cells there, a lot of infection, if you don't regulate the killers, there is massive immunopathology. So initially it's coming up to really regulate that. And during that early phase, doing PD-1 blockade actually can cause excessive damage. So this PD-1 pathway comes up to essentially to tame the killer cells, if you want to put it that way. Okay? But in an acute setting for memory, really PD-1 is gone and doesn't play much of a role in what happens during memory formation after acute infection.
Good. There is another one from the technology point of view. Nabil Ahmed asking, can we generate CD8 stem-like cells by iPSC, and can they be then used for the therapy?
Yes, absolutely. I think many people are trying to do that. People who do T-cell therapy are trying to see if, based on the gene expression program that we and others have defined, including the key genes in there. Yes, I think that this, for T-cell therapy, these ideas are being already tested. Yes.
Very good. So you actually showed a lot of, you showed a slide of the AD1 therapy in various types of cancers. So MF Khan wants to know whether it has also a role in infectious mononucleosis.
Yes. So
AD1
you're
therapy.
talking about the EBV-induced infectious mononucleosis. And I would say that it might be dangerous because during that acute phase of, during this happens early on in certain people when you have very high levels of EBV. There also a lot of CD8 T cells at that time, which are being restrained. There's a lot of PD-1 expression on those EBV-specific cells at that stage. And it would be a very risky thing. There might be much more excessive damage if you do PD-1 blockade. So it's a balance at that time. So one of the reasons why PD-1 has not been used that extensively in chronic viral infections really is because of the concerns with excessive immunopathology. And one of the places where it is being considered now is hepatitis B, because especially in the hepatitis B carriers, because there really isn't a good antiviral. But again, there will be concerns about excessive liver damage.
Right. So the other question from Arinda, I think that has already been answered about the iPSC and all that. Komal Sharma wants to know how to combat resistance to ICIs. I don't know, what does she mean by that?
Yeah. He's talking about immune checkpoint inhibitors, how to, yes, that's a very good question. Yes. So as I mentioned to you, although PD-1 is very effective, many people don't respond. And then some who respond will not continue to respond. So that's what he's talking about, resistance. And there are many potential mechanisms of resistance that other people have defined. One is that very often tumors undergo mutations in beta-2 microglobulin. So that essentially the antigen presentation is not very effective. Uh, so that epitope is not being expressed very effectively. Other means of resistance mechanism is less responsiveness to interferons, interferon gamma. So the tumor cell often adapts to evading the CD8 T cell response. It's like you get viral escape, you get a CTL escape. Here, the tumor is escaping by essentially interfering with either the MHC presentation pathway or processing pathway, or by other mechanisms. So this is a challenge in the field that I think one will need to overcome.
So question from Michael: why do we use mice only for understanding immunology? Mouse doesn't.
I'll let Vijay answer that later.
Well,
we use mice for a very good reason, because you can do mechanistic studies.
yes.
Yeah, because you have all the reagents. You have the knockout mice, you have the knock-in mice, you have transgenic mice. You have all the reagents for doing. You can only understand mechanisms in mice.
Genetic.
And sometimes those hold up, but very often, I would say almost always, the general principles that have been defined in mouse studies have always held up in human studies. The details sometimes are different, but the principles, I can't think of any principle defined, immunology principle defined in mice that has not held up, because that's where the mechanistic studies are done.
And also you can be called a mouse doctor and MD.
Absolutely. I'm very happy to have that honor.
What is the role of stem-like T cells in infectious diseases like mycobacterial infection? Do you think they would have a role?
Yes, I think again, there's something. I think anything that is chronic would have. There's also possible that the CD4 cells will have similar pathways. And again, there's papers that are coming out which is kind of alluding to some similar programs in CD4 T cells. I think chronic antigen, the microenvironment, the cytokine environment created, I haven't mentioned myeloid cells, but I think the myeloid cells are playing a very big role in these differentiation pathways and also in sometimes resistance to checkpoint. So it's an interplay between myeloid cells and the lymphoid cells in many of these things. But chronic antigen is the main driver for giving you these cells.
Rafi, I will just take one more last one, and that's from he has asked two, but I will only take one of his. Okay. Whether blocking PD-1 would promote CD8 effector memory responses.
Well, again, I told you that under chronic conditions is when you have PD-1 expression on the T cells. When you do that, yes, of course, you get more of the effector-like cells that I've described. But if you do the right combination, that was my main message from the combination therapy. I don't mean to imply that's the only way of getting better effector cells, but the IL-2 is really in our hands of the many things we've tried by far the best. So this combination therapy, that's what it's doing. It's giving you much better effector cells, transcriptionally and epigenetically different. And I think that those might actually give you better memory cells after the tumor is cleared or after the infection is cleared. I think that pathway has been altered. And that's why we're excited by that finding, because I didn't have time to go into it. But now you're getting, under normal conditions, when you do the PD-1 blockade, as I showed, TIM3 comes up, effector program comes up, but those cells, again, are not very long-lived. They don't have TCF1, which is needed for good memory cells. They don't have very high levels of CD127, so they don't survive very long. So these key things are missing, but what the combination therapy is doing, really amazingly, is we now get this effector population, which has the potential to become long-lived memory cells because they have TCF1. So we now have granzyme B positive, TCF1 positive cells, and TIM3 is low on these cells. So this is giving you an effector population which potentially could result in more long-lived memory cells.
Of course, you said, of course, I've seen that there are quite a few more also in the panelist column, but I will pick up one or two. People in the autoimmune field. Of course, you said that the CD8 memory stem cells formed in everything, but they want to know whether CD8 memory stem cells are also found in autoimmune.
I will save time and defer that question to Dr. Kuchroo, who's
has been
in autoimmunity. Anything he says about autoimmunity will have more credibility than I will. So let me just pass on that question. But I'll take another one if you have it. Yes.
You should stay on, and then we can take this up during,
Yes, I'm looking forward to hearing Vijay's talk. So
we have been right on the dot at 7 o'clock for our time. Thank you so much for this wonderful talk. It was really great and so much to learn from this talk. Thank you so much.
You. Always a pleasure. Thank you.
Yeah. Okay. Okay, friends, we move on.
I'm going to mute myself now. I'll mute myself.
You have to shut up now. So
yeah, yes, I will mute myself. Listen to my friend Vijay. Yeah.
So our next speaker, and in fact, the last speaker, is Dr. Vijay Kuchru. I'm sure everybody in this audience knows him. A very handsome-looking immunologist around the world. He is the Samuel Wasserstrom Professor of Neurology at the Harvard Medical School. He's a senior scientist at Brigham and Women's Hospital. He's a co-director of the Center of Infection and Immunity, Brigham Research Institute, Boston. He has been the boss of Amit Avasthi. And of course, his laboratory has been engaged in a range of activities from autoimmune diseases to describing for the first time the TIM family of genes. His laboratory is also associated in studying cancer immunotherapy, but the first to describe the development of a very highly pathogenic T17 cells, which have been shown to induce multiple different autoimmune diseases in humans. He has 25 patents. He's founded 5 different biotech companies. including the co-stim pharmaceuticals, Potenza pharmaceuticals, Tizona pharmaceuticals, Celsius pharmaceuticals, and go on and on. He is on the advisory board of several sort of internationally recognized companies, and of course in institutions. His CV is so very long. I don't want to really spend too much of time on that. I don't see him. Vijay, where are you?
I'm here.
I've been talking about you. There you are now. Now you're wearing a hat. I mean, look at this. Oh my God, you lost them all.
All right.
19 or what? But anyway,
All right.
the floor is yours, and you are really going to speak on a very important topic of checkpoint molecules and induction of anti-tumor immunity. So you know that we are 45 minutes for you, and then there will
Yes. All right. If you stop, then I can start talking. Thank you very much for that wonderful introduction, and thank you, Rafi, actually, to setting the stage. I really want to actually thank Sunil and Amit and Narendra for inviting me. I often seem to rush at the end, so I really want to acknowledge people who did the work. In fact, I will talk about TIM family of genes, and TIM3 was the founding member, and Laurent Monnier and Catherine Sabatas were the initial discoverers of this family in my lab. And then a lot of people who did followed up, the Chen and Manu and Norio, Asaf, Taka, Yasuhiro, and Karen. And a lot of work actually was done with Arlene Sharp, who did most of the transgenic and knockout mouse I talk about. And one of the first actually descriptions of a role of TIM3 in T-cell exhaustion, in fact, was in HIV. And then I contacted and actually Rafi, and Rafi and I have a very nice paper showing that TIM3 have a very important role in T-cell exhaustion in chronic viral infections. And we have continued on to study TIM3 and the family of genes, and Curie and Chow and Lionel with Anna Anderson, who was my student and a postdoc, now a faculty member, has continued on studying the role of TIM family of genes in cancer. A lot of work on single-cell RNA-seq. This was done together with Aviv Rego at the Broad Institute, where I'm also a member. And Orit, Nir Yosef, and was were joint postdoc, and Maramit was a joint postdoc as well with Aviv and myself, who did most of the work. So I just have to tell you something that the Catherine, who cloned initially the gene in the lab with me, is now at Novartis, and the anti-TIM3 antibody is actually now called as sabatolimab. And it's actually very interesting that a student who cloned the gene went on to do the clinical trials, and the antibody is named in her honor. So just to take you along, what my outline is, I will introduce, in fact, the co-inhibitory molecules. And this is just to wrap it up, since it's a course and there are different levels of people with their background. So I'm going to actually briefly talk about checkpoint molecules, the co-inhibitory molecules, and how they has been used for cancer immunotherapy, how we discovered a TIM family of genes, and together with many investigators, including Rafi, showing that it has got a role in TIM3 as a role in T-cell exhaustion. And the last one is immunosuppressive cytokine, IL-27. In fact, type 1 cytokines and IL-27, in fact, cooperate in humans to induce these co-inhibitory molecules. And I think that's what Rafi was mentioning, that the environment in the tissues may be playing a very important role in how we induce T-cell exhaustion. So, in fact, the cancer immunotherapy is not new, and there has been a whole history of what cancer immunotherapy can do. In fact, there were indications 100 years ago, first showing by Workhoff that the tumors are infiltrated with immune cells. And then there was actually a toxin, which was a bacterial toxin, which was generated and showed that it may have some impact. And then Burnett, in fact, proposed the hypothesis of immune surveillance And BCG in 1976 was used for the treatment of bladder cancer, IL-2 as cancer immunotherapy, and there was adaptive therapies and T-cell therapies. And then till we reached to 2011 and 2014, when we really started using, based on science and basic science, we started blocking these inhibitory receptors, what we call checkpoint molecules, and that have major impact in cancer immunotherapy. So immunology has offered hope for over 100 years that they're going to actually inhibit, prevent, and or cure cancers. And obviously 100 years, in fact, not much happened. And being at the Broad Institute, and they spent all their effort in genomics of tumor cells because there was these heated discussions that immunology has not offered any hope for cancer therapy. But things are different now. In fact, there's a new strategy, as Rafi mentioned, that we have these inhibitory molecules. We used to call them co-inhibitory molecules, and they are, and the cancer immunologists started calling them checkpoint molecules. And this is the molecules that we normally come on T cells to induce T cell exhaustion, and blockade of these molecules induces very enhanced T cell responses to cancer, and in fact, in many patients, cure the cancers. So I'm going to go to the basics, and Rafi mentioned it, just to define basic principles of T cell activation. T cells obviously have a T cell receptor, and peptide MHC complex provides signal 1, which gives specificity to T cell activation. And Mark Jenkins and Ron Schwartz first described that that is not going to enough to put T cell into activation, expansion, or differentiation. And they showed the data that it needs a second signal, and which we call co-stimulation now, and that the signal 1 and signal 2 now is optimal to put T cells in cycle. They activate, expand, differentiate, acquire effector phenotypes, and then they have these inhibitory molecules that they come, and then these inhibitory molecules actually turn them off. And here's an example: is peptide MHC complex providing signal 1, B7-CD28 provides signal 2, and then once the cell is fully activated, CTLA-4 is one of the examples of inhibitory receptors. It comes on the T cells and it turns the T cells off. Otherwise, will induce IL-2, the T cells will expand, and then you have, once the T cells are activated, you upregulate a whole series of inhibitory molecules. And here is the CTLA-4 is one of the example. It induces T cell inhibition. So an optimal activation and inhibition is required for maintaining homeostasis. And after the CTLA-4 was discovered, and we have now discovered a whole series of activating receptors, PD and then they acquire these inhibitory receptors, CTLA-4, PD-1, TIM-3, LAG-3, TIGIT. And these cells are not the lame ducks. They also make these inhibitory cytokine IL-10. And with the result, now you are turning off the immune responses so that you have optimal activation, effector function, and clearance of infection. And then you bring all the cells to homeostasis by expressing these inhibitory receptors. And the tumors, luckily or unluckily lucky for them, is actually have co-opted this whole system. And here is, I'm giving an example, is that if you transplant a tumor into a mouse, the tumor progressively grows. It's not because the immune system is not reacting, that CD8 T cells will infiltrate the tumor. And in fact, if you analyze CD8 T cells and you see that the cells initially will have no PD-1 or TIM-3 on them, or they will acquire PD-1, or they will acquire both PD-1 and TIM-3. As they acquire these inhibitory receptors, and they can see they progressively lose their effector functions. In fact, you can see that when you don't have the TIM-3 or PD-1, they are the ones that produce most of the cytokines and effector functions. And my computational biologists call them wimpy because they upregulate PD-1. They don't lose all the effector functions, but when you have both TIM-3 and PD-1 together, you become almost dysfunctional, losing all of the effector functions. As I said, we used to call these molecules that co-inhibitory receptors or second signals, negative signals, and they have been renamed or rechristened as checkpoint molecules. So I'm going to briefly touch on the PD-1, which has revolutionized the way cancer immunotherapy is working. So what you have is, as I said, that you have peptide MHC complex, and you class 1 molecules are expressed onto tumor cells. They will upregulate T-cell receptor, and these are activated T cells. And the CD8 T cells will produce cytokines like interferon gamma, and this interferon gamma will act on the tumor cells to upregulate PD-L1, and PD-L1 will inhibit the CD8 T cells and inhibit their effector functions. And lots of antibodies. In fact, last I counted, and there are already 4 anti-PD-1 antibodies approved by FDA. And there are over 100 different anti-PD-1 antibodies being generated and are being tested right now. And this is just one example of what Rafi was talking about. And Rafi should get the credit for, in fact, suggesting that the PD-1 is a key molecule that cooperates with CTLA-4 to induce T-cell dysfunction in chronic viral infection. And that happens to be true also in tumors. And here, anti-PD-1, when you, in fact, block the PD-1 signaling, and this is a spider plot in a melanoma patient showing that you can see that some patients will not respond. But if you give anti-PD-1 therapy, and you can see the tumors progressively lose, there are regress. And here are at the beginning of the therapy, you can see this by MRI, you can see all these tumor metastases. These are sickened tumors, and within 12 weeks of treatment, and these tumors are gone. And with the result, what happened was that the tumor immunology from being on the fringes actually came to the forefront. It became very exciting because many of these therapies, in fact, the PD-1 anti-CTLA-4 was based on actual basic research that you have activating receptors that will put cells in cycle, but when you block the inhibitory receptors, which are co-opted by tumors, and when you block them, and you can invigorate the effector T cells, especially the stem-like cells that Rafi talked about, and you put them in cycle, and you can induce anti-tumor immunity. But here is the question. The question is that when it works very well, and there are durable responses. But the problem is only 20 to only 30% of the patients will actually respond to anti-PD-1 blockade. If you block both CTLA-4 and PD-1, it can maximally go up to 50%. And that raises a very simple question. If they're all tumors, they have PD-L1 them, and the effector T cells express PD-1, why don't they all respond to anti-PD-1 therapy? And this brings me to the next part of my talk. So what I told you is for T-cell activation, you don't need just one signal. T-cell peptide MHC complex is signal 1. Then CD28 is a key molecule that puts these cells into cycle. It activates and expands and differentiates these cells. And these cells express now inhibitory receptors to bring them to homeostasis. Otherwise, every time you have a bacterial or viral infection, your lymph nodes will actually swell and they will not come back to normal. And tumors, for some reason, and I'll get into it, have co-opted these inhibitory molecules. The moment effector T cells go into the tumors, they will upregulate the series of these inhibitory molecules. I gave you an example of PD-1 and TIM-3. And now cells can't go and evoke full potential. And blocking this PD-1 pathway has been shown to induce durable responses in about 20 to 30% of melanoma patients. And this therapy has been now approved for 7 different cancers. But the question really is that if PD-1 comes on all effector T cells, why don't all patients respond? So this brings me to there are other inhibitory receptors. I gave you examples of 7 of them. And I'm going to actually go into how we discovered TIM3, and TIM3 and PD-1 will cooperate together to give you inhibit anti-tumor immunity. And as I said, that it's a molecule that together with Rafi, we showed that it's very important for T-cell exhaustion. So what we did was we were looking for molecules that are expressed on effector T cells, especially Th1 cells, because Th1 cells induce autoimmunity and tissue inflammation. I'm keeping an eye on my clock here. Because they induce effector functions, and they go to traffic the target organs, and the target, they get reactivated, produce cytokines, and induce tissue inflammation. And we knew that Th1 cells are very critical. So we had made a T-cell receptor transgenic mouse, and we were trying to actually identify monoclonal antibodies that will bind to Th1 cells. So we were immunizing rats, and what we did was we immunized rats to induce monoclonal antibodies. In fact, we generated something like 20,000 different monoclonal antibodies. And we generated at least not one, we generated 4 different monoclonal antibodies, and 2 of these antibodies were identifying this molecule called TIM3. And as you can see, the TIM3, in fact, stains all the Th1 cells. It doesn't stain Th2 cells. It also induces, stains TC1 cells, interferon gamma producing CD8 T cells, but not the TC2 cells. We went ahead and cloned the gene, and this was in collaboration with Gordon Freeman. And when we cloned the gene, it was a novel gene. In the database, it had an IgV, it has a single peptide, IgV domain, mucin domain, and the long tail, and long tail has 6 tyrosines in it. And there is about 69% homology between mouse and humans. And this also resulted in the identification of this whole family of TIM molecules. And there are 8 molecules in mice, and there was a reverse evolution, and there are only 3 in humans. And it is at a very, very interesting place in the human genome, 5q33, which has been associated with a number of autoimmune and inflammatory diseases. It's also on the centromeric end of the mouse chromosome 11, which again has been associated with atopy, allergy, and asthma and autoimmune diseases. And we're going to talk only about TIM3. And we wanted to know what the function of TIM3 is. We took the external domain or IgV domain of TIM3, and to increase its half-life, we put it on the Fc portion so that we have a soluble TIM3. And we injected in vivo, and this was the molecule that we also collaborated with Rafi to see what its function is in T-cell exhaustion. And so we were trying to look for the function, but more importantly, we wanted to identify the ligand. And we went ahead, and what we found was that the soluble form of TIM3 was in fact binding to lots of tumor cells. Here's an example of a CD8 tumor called TK1. You can see the soluble forms of TIM3, both the IgV domain only or full-length form was binding. And Chen Zhu went ahead to find the ligand. And you can see that when he did a mass spec, IP mass spec, there was this blob of a highly glycosylated protein. When he deglycosylated it, it was a very defined protein of 30 kilodaltons. He did a mass spec and identified and told me that it was galectin-9 as the ligand for TIM3. And in fact, very first time he told me it was galectin-9, I did not believe him. And in fact, I had a very interesting idea that the ligand for TIM3 must be IgV-like domain. It will bind to each other and inhibit or activate T cells, but this shattered my dreams. I went and told him to go ahead and in fact further sequence it. And I made him to do this experiment 5 times, every time picking up a different part of galectin-9 and such that there was an 18% coverage, I had no reason not to believe that the galectin-9 was not the ligand for TIM3. In fact, I'm not showing this picture, is that the galectin-9, when it binds to TIM3, it induces aggregation, and it aggregates TIM3 positive T cells, and then the cells start actually popping or dying, and it looks like almost like a Diwali or 4th of July, where you have these fireworks if you're ever watching these T cells together with galectin-9. So we came to the conclusion that the galectin-9 TIM3 interaction was an inhibitory signal inducing cell death of the Th1 cells. If you block the pathway in vivo in an immunized mouse, and you can see it enhances interferon gamma and IL-2 production. And if you ever gave it to a mouse undergoing an autoimmunity or type 1 diabetes, and it exasperates, in fact, the disease, autoimmune disease. So we decided at that stage that it is an inhibitory molecule. And I'm going back to this cartoon. What is its role in anti-tumor immunity? So when you transplant the tumor and tumors progressively grow, they have infiltration of these T cells, CD8 T cells, and the CD8 T cells will start expressing these checkpoint molecules, and which turns these T cells off, and they become dysfunctional. And in fact, if you take out the CD8 T cells from the tumor and you're staining now for TIM3 and PD1, you have some cells, a very small fraction, that do not express either PD1 or TIM3. There is a fraction that will express PD1 alone, but about 50% of T cells will express both PD1 and TIM3 together. And if you look at the cytokine production from these cells, if you took the TIM3 PD1 double negative cells, they will produce IL-2. This is intracellular cytokine staining, TNF, interferon gamma. The moment you express PD1, you will lose some of the IL-2 production. You may lose TNF production, but not all PD1 positive cells are losing all effector function. If anything, you can see there is increase in interferon gamma production. We think this must be because there are some of the recently activated effector cells. But when you have both TIM3 and PD1 together, you lose almost all of the effector functions. So I'm going back to this concept that Rafi published and talked about, is that there is this antagonism between the stem-like cells that express TCF1 or TCF7 is the gene, TCF1 is called the protein. And in fact, these are the stem-like cells that respond to immunotherapy. But the moment TIM3 comes, they lose the expression of TCF1, and these are the cells that become exhausted. And you saw this from Rafi, there is some sort of antagonism between the TCF7 expression and TIM3 expression. And this is totally unpublished data. So we infected T cells, naive T cells that we know already have TCF7 in them. And then we expressed in them TIM3 that we can induce by doxycycline treatment. And we wanted to see that if you force expression of TIM3, what happens to TCF7 expression? And in fact, something that Rafi showed, but there must be some active process by which when you increase the TIM3 expression of T cells, in fact, you can see progressively TCF7 goes down in these effector T cells. So there is this antagonism. And in fact, there are two molecules that were identified and that are expressed on terminally differentiated T cells, and they show antagonism between TCF1 expression, and that's TIM3 and CD39. And we don't see this function of this inhibition of TCF7 or TCF1 with the CD39, but we see this TIM3. And we are figuring out what is this antagonism between TIM3 and TCF7. So just going back to cancer immunotherapy for a minute, and then the question was what happens and if you block. So these are the mice that have tumors on them, and you give anti-TIM3 or anti-PD1 or PDL1, and you can see the isotype controls don't work. Anti-TIM3 alone doesn't make any difference. PD1 sometimes works and sometimes doesn't work. But if you co-block both of these inhibitory molecules, and this is actually inducing complete inhibition as if the tumor melts away. And this is the reason why anti-TIM3 is being tested. There are at least 12 different clinical trials for co-blockade of TIM3 with PD1. and resulting in fact, there's encouraging clinical data that's coming out in solid tumors. But I'm going to show you a couple of slides where anti-TIM3 or sabotuzumab is actually showing beneficial effects, and that's acute myeloid leukemias. And this cytabine is an demethylating agent, and which is normally actually treated used to treat acute myeloid leukemia or myelodysplastic syndrome. TIM3 is highly expressed on acute myeloid leukemias, and this is the standard therapy is to give this cytabine. And I want to emphasize that it's such a bad tumor that there has not been a single approval in last 10 years for this therapy, for this tumor. And patients, 80% of the patients will die within 5 years of the diagnosis. So the anti-TIM3 has been tested alone, either or with a chemotherapy decitabine. And this is really a very small trial. This was a safety trial, which has now been extended. And you can see that if you give anti-TIM3 antibody in the high-risk myelodysplastic syndrome, and you can see wherever you see yellow, there are these patients showing complete responses. And in fact, some of the patients have gone on to for 2 years without relapse. And if you have orange, you don't only have the blood responses, but you also have complete responses in bone marrow. If it's a gray, and they have the stable disease, and if you have black, and this is why the relapse happens. And just to summarize, and I don't want to, it's only a very small trial, and there are now hundreds of patients have been added into the trial, but this makes a point, is that if you have high-risk myelodysplastic syndrome or completely refractory AML, acute myeloid leukemia, or if you have acute myeloid leukemia first-line therapy, and if you give anti-TIM3 sabotuzumab into these patients, you get between 50 to 60% response rates. So just to sum up the second part of the talk, what I told you is that the PD-1 and CTLA-4 are not the only inhibitory molecules. And we identified TIM3 and TIM family of genes by making monoclonal antibodies against Th1 molecules, Th1 cells. And it's a whole family, and TIM3 is the cheerleader. And together with Rafi and then with Anna, we have shown that the TIM3 cooperates with PD-1 to induce T-cell exhaustion, and co-blockade of TIM3 and PD-1, in fact, inhibits the development and retards tumor growth. And in fact, I showed you that the data I showed you is that anti-TIM3 antibody induces durable responses in these acute myeloid leukemias and myelodysplastic syndrome. How am I doing with the time? I have a last.
You have 15 minutes more.
Okay, wonderful. So what I also actually showed you in the second part of the talk is that the Rafi in his paper had shown, and he talked about it, is that you have stem-like cells that have TCF1 or TCF7 expression, and they are the ones that give you, the stem-like cells will give you this burst when you do PD-1 block, and they are the ones responding to therapy. But the moment TIM3 comes up, we are clearly seeing that even with the forced expression of TIM3, you are losing the TCF7 and you're losing stemness. And then the cells are on their way to become effector T-cells. And if you engage PD-1 and TIM3 together, and you are inducing T-cell exhaustion, and co-blockade induces actually retarded tumor growth and induces anti-tumor immunity. Now, Rafi actually mentioned this, and we were curious what induces TIM3, and that may have a wider application. So we identified an immunosuppressive cytokine called IL-27 as a key molecule for induction of TIM3. So Chen Zhu did the screen, and he took all the cytokines and he activated both CD4 and CD8 T cells. and then looked at what cytokine induces TIM3 expression. And this is data is published. I'm going to go through quickly. And one of the cytokines that was inducing TIM3 is IL-27. What was interesting in this first set, we didn't have type 1 interferons. And in fact, a series of experiments we had done with David Heffler, we find that type 1 interferons, either alone or in combination, will induce even more TIM3 expression. And you can see that this is qPCR. And then this is at the protein level. I told you that Th1 cells will express TIM3 expression. This is a FACS analysis compared to the unactivated or without differentiation. And when you put IL-27, a vast majority of Th1 cells will induce TIM3 expression. So IL-27 is a suppressive cytokine, and it comes during a resolution of inflammation. It has got two chains, EBI3 and P28, and it has got a unique receptor called IL-27 receptor, which collaborates with the GP130, which is an IL-6 receptor. And the two together will form this signaling chain, and these heterodimer induces the inhibition of inflammation. And we knew about this when Amit was in the lab. In fact, he did a lot of work on the role of IL-27 and showed that the IL-27 was a key cytokine in suppressing inflammation in inducing IL-10 producing T cells in the lab. So we were interested in seeing how does IL-27 actually go and act on naive T cells and induces IL-10 and immunosuppressive phenotype, and you can actually activate the T cells. And at the end of it, you can see IL-10 and all the relevant transcription factors and cytokines get induced. But we were not interested in actually looking at the end. We wanted to know, how does a cell march across in 72 hours to give you this phenotype? And what we did was we did RNA-seq analysis, and it may have been.
So this was, see, we know as much about immunogen design as.
I can barely hear you. Okay.
Please put your question in the question box. Do not interrupt kindly.
Okay.
Go on, Vijay.
Okay. So what happens is that if you took the naive T cell, you put an IL-27, we collected RNA-seq data sets, and it may not have been RNA-seq. It probably was it was a chip analysis, but a whole genome analysis, 18 different time points. And what we find is that they form this chain network, and it's going to be published soon. And but Asaf Mahdi actually did this analysis, and you can see there are over 1,400 genes that are induced in T cells when you have IL-27. And IL-27 receptor knockout mice actually don't see induction of genes. And then if you actually have Th0 cells which don't have any cytokines, and they look like the IL-27 receptor knockout mouse. As he was analyzing the data, and he actually honed on, and because the other part of the lab was working on TIM3 and the exhaustion, T-cell exhaustion in cancer, he found there's a set of these genes that were coming together. And you can see they were induced in wild-type T cells, but not in IL-27 receptor knockout mice. And you can see the whole series of genes, the CCAM, TIGIT, LAG3, TIM3, and they were all coming together as a module, and they were induced by IL-27. So IL-27 looks like not only induces IL-10, which is here, but is also inducing a whole series of checkpoint molecules. And I'm coming back to this picture that you've seen before, and you can activate T cells with the activating receptor. Then you have this whole series of checkpoint molecules, and we are actually seeing that IL-27 may be inducing these inhibitory receptors on the surface of these activated T cells. And the question was at that time, and I asked Anna Anderson, who was in the lab, I asked them that, do these molecules all come on the same T cell? Or all they express, some express TIM3, some express PD1, some express CTLA4, and then when you do population RNA-seq, it looks like that they are together on the same cells. So the one way to figure out is to do single-cell RNA-seq, and we, in fact, did this have been doing this for 6-7 years now with the Aviv Regev at the Broad Institute. I don't want to go into the details of it. And the data is published. And if you look at CD8 T cells, and this is the single cell data set, and this is the Pearson correlation coefficient showing which are the molecules coming together. You can see TIM3, CTLA4, TIGIT, LAG3, PD1. In fact, PD1, TIM3, LAG3, and TIGIT, the correlation coefficient on a single cell to come together is 10 to minus 23. That means these cells, these molecules are not coming on their own. They are coming as a module, and they are co-expressed and they are co-regulated. And same thing is true with CD4 T cells. If you do single cell RNA-seq, and you can see all of these molecules, LAG3, TIM3, TIGIT, and CTLA4, PD1, they all come together. And interspersed with these inhibitory molecules are these activating receptors of TNF receptor family members, GITR, OX40, 41BB, and those must be the right receptors to agonize these T cells. So it actually brings that to a question that is this only in the RNA-seq level? We also did CyTOF. Again, on the CyTOF at the protein level, you can see that all these molecules are coming together. You can see, in fact, we are finding new ones too. The TIM3, LAG3, PD1, TIGIT, they're all coming together. Interspersed are these activating receptors. So if you look through biology, through the organism, that every time you are trying to repress, even from the Drosophila onwards, that if every time you're trying to repress a cell and inhibit its function, what you do is you want to activate it to keep the organism alive, and the activating receptors come up. So these must be the right kind of activating receptors that are antagonizing inhibitory function. And these may be the ones that should be actually agonized in cancer immunotherapy, and that's exactly what's being done. So what I told you is that these molecules are not coming actually willy-nilly. In fact, in each cell, PD1, TIM3, LAG3, and TIGIT form this module, and they are co-expressed at the single cell level. And this is, I'm just showing a couple of pictures of the CyTOF, and this is the CyTOF analysis. It's almost like a multidimensional fax. And just to orient you, this is CD4 T cell. These are the CD8 T cells. And the moment I saw it the first time, it reminded me of an elephant. I grew up in India, so all I can think of elephants. And you can see that this is the head, and this is the body, this is the trunk, and these are your legs. And you can see that the checkpoint molecules, the PD1, TIM3, LAG3, and TIGIT, they're all actually staining the elephant's head. And if you look at whether these are being expressed by IL-27, what IL-27 has in role, and the wild type T cells are blue, and the IL-27 receptor knockout derived T cells from the tumors are in red. The only place IL-27 receptor knockout can stain is in elephant's head. And in fact, this is where the checkpoint molecules are. So IL-27 as a cytokine is inhibiting the induction of PD1, TIM3, LAG3, and TIGIT. And I can tell you that it doesn't do a complete job, and suggests that there must be other cytokines that are cooperating with IL-27. And we find that type 1's interferons are critical, which come during chronic inflammation. And if continued production of type 1 interferons will collaborate with these molecules to induce almost complete T cell exhaustion, with IL-27 induce complete T cell exhaustion. So what I told you, IL-27 as a cytokine will induce PRDM1 and CMAF, two transcription factors, and they will actually induce this whole module of checkpoint molecules, PD1, TIM3, LAG3, TIGIT, CTLA4, and also some of the novel molecules, and engagement of these molecules will induce T cell exhaustion. And I have this picture we took the data sets from human tumors, and you can see non-small cell lung carcinoma, colorectal carcinoma, melanoma, and hepatocellular carcinomas. And these are the genes, single cell RNA-seq genes that are identified. And you can see there's a core of genes, 18 genes that are common on CD8 T cells in these tumors. And you can see this core module is in fact expressing TIM3, PD1, CTLA4, TIGIT, LAG3. I want to apologize that I didn't tell you that this is the gene name for TIM3, HAVCR2. So it suggests that not only in mouse, but in humans form this core module of this checkpoint molecule. So I told you that this is the cytokine, and it collaborates with type 1 interferons to induce PD1, PRDM1, which Rafi also talked about, with and CMAF, the two cooperate to induce module of these genes. And what we are doing now is, in fact, we know the cytokine, receptors, and we know also the transcription factors and all the transcription machinery. And we are actually using a very interesting computational trick. It's called Steiner tree algorithm. And in fact, my postdoc, Nir Yosef, wrote this algorithm called ANAT, whereby we can actually build this network and protein-protein network to go from the transcription all to the receptor, and building all the major nodes and subnodes that get activated by IL-27. So this paper was published, I think, a year ago. And if you want to see it, you can go ahead. Actually, it's in Nature with all the data. So what I told you is that the antigen-presenting cells normally produce IL-27 during resolution of inflammation. IL-27 will act on effector T-cells. And not only does it, in fact, induce IL-10 production, but it also induces a whole series of these checkpoint molecules on effector T-cells, and to induce dysfunction and ultimately death. And we are actually looking at who produces IL-27 in the tumor. We know it's being produced. We have just made a reporter for IL-27 for both P28 and EBI3, and we are beginning to identify the cells, and we think the tumor-associated myeloid cells and stromal cells may be the key source of production of IL-27 in the tumors. So where are we going? We are not actually setting at the one single cell level. In fact, we are taking this and both the spatial transcriptomic analysis at single cell level, and we're going to put this whole machine together to see what happens in the tumors. Is we can in fact tell you that what are the cells that are critical for induction of T-cell exhaustion. We can tell you the molecules, whether it's the cell surface or soluble molecules, what do they induce? How are these cells coming together? And what are the partners that may be impacting? And we can in fact tell you the machinery that's being activated to induce the molecules and cell surface molecules that are adhering together to induce a T-cell dysfunction or exhaustion, and what needs to be regulated and what molecules are produced by T-cells to perpetuate this T-cell dysfunction. And in fact, this will be something that will be useful as an atlas, as a machinery, and targets that from which we can identify not only the cell types, not only the soluble molecule, but the cell-cell interaction at the tissue level, how at T-cell and immune cell and tumor cells are interacting together to induce T-cell dysfunction, and how can you in fact use and target them to induce and promote anti-tumor immunity. So let me conclude what I told you, that the activated T cells express inhibitory receptors to turn these T cells off. What I also told you is that the blockade of CTLA-4 and PD-1 induces tumor regression, but we have the whole, they don't do a complete job, because at best between 30 to 50%, even when the co-blockade will induce tumor durable responses or tumor regression. And that results got us into, are there new checkpoint molecules? And I think TIM3 is one of the novel molecules that expressed on the effector T cells. And what I have not told you is that it's also expressed on myeloid cells. And we can tell you that TIM3 is co-expressed with PD-1 to induce define deeply exhausted T cells. And that's because it antagonizes the expression of TCF1, and blockade of these two molecules induces almost complete tumor regression. And I showed you the data that IL-27 coming from tumor niches is in fact inducing TIM3 expression. And in fact, it doesn't induce only TIM3 expression, induces the complete module, and which you may call exhaustion module, which is actually a misnomer, people call it. that there's a module of these inhibitory receptors. And I showed you the data that TIM3, PD1, LAG3, and TIGIT have a Spearman correlation of 10 to minus 3, and these cells also induce IL-10. And this is my lab. We have a soccer team. We have not won many games, but we still are enthusiastic. And we call ourselves Kuchru Lions. And after losing all these games, and I asked Alex Schnell, who is the captain, whether she could consider changing the name of this team. And she says no. I thank you for your attention. I'm happy to take any questions you may have.
Thank you very much, Vijay, for the wonderful talk. I'm sure there are a number of questions in the question box. I have a couple of them. I just want to know, of course, you know, the TIM3 and PD1 are also expressed in COVID-19, you know, where the T-cell exhaustion is one of the issues over there, and they're both expressed over there. How do we bring back exhaustion back to normal?
I think Rafi may have ideas about it, but it's very clear that once you have TIM3 and PD1 both upregulated, and if the receptors are engaged, and there are changes, epigenetic changes, and that it's very difficult to bring them back from that state. There is a transitory state, I think Rafi could talk about it, is that where you could potentially bring them back. But once they're terminally exhausted, it's very difficult. Rafi, do you have any ideas, suggestions? You're on mute.
Yeah, thank you, Vijay. Yes, I agree with what Vijay said. Where you can manipulate the cells most effectively is really at the stem cell, because when these cells are then differentiating, you can really change their path, as I showed you with IL-2, but I'm sure there are many other ways of changing that path. Also, the transitory cell may be a little bit still amenable to some of these changes, but once you have gone beyond that, that's a difficult cell to reprogramming that cell is difficult. And I think that will probably require some chromatin modifications.
And Vijay, I wanted to ask you, of course, you know, the slide that you showed, there are a lot of molecules, actually, many of them are activating, and the similar number is also inhibitory. And yet we find that in immunotherapy of cancer, it's only effective in a very small number as yet.
Yeah.
So one was thinking in terms of, do we have a variety of cells? Do we have heterogeneity? But you were saying that every T cell has this. So
yeah. So it's a very good question. What you raised is a very good question. I raised the question, didn't answer it. So first one is that these cells have the potential to express all of these receptors together as a module. And if you block one, you may not effectively induce activation of the T cells. And in fact, there's a data already coming out showing that in the PD1 resistant trials in lung cancer, and there is TIM3 being expressed very highly. In other places, TIGIT is being expressed highly. So you may have to block multiple receptors in order to invigorate effector T cells from those transitory states. That's number one. Number two is that you can activate T cells, but you are going to hit a ceiling beyond which you can't do anything, because you have to make the tumor amenable to effects of T cells. Just imagine if the tumor is covered and with a fibrous mass, or is producing massive amount of suppressive cytokines, the effector T cells you generated will not either get in or will get actually turned off immediately. So something else has to happen. So we will soon reach a ceiling beyond which we will not be able to actually mediate an effective effector function with just having T cell mediated therapies. And I think Rafi actually touched on it, that keep an eye on myeloid cells. What are they producing? What are they doing? And I talked about the fibroblasts. They're making a shield, and the shield is producing with this, our reporter mice are producing IL-27. So even before you get in, they are actually inhibiting you. So I think multiple and combination therapies has to be not only those that activate T cells, but impact other cell types as well.
I think you have answered this question also from Shivnath. He wanted to know the same thing. If both activating and inhibitory come up together, then what decides whether T cell will be activated or inhibited in the end?
Yeah, I think it's a combination of things, obviously, that you need to have activating receptors, put them on cycle. And I want to also emphasize, superficially, they look all the same, that they are inhibiting your effector function. But they each have their nuanced effects. And actually, we talked about one of the molecule, protoplanin and PROCR, they're antagonizing IL-7 receptor functions. And this is what Rafi showed, is that as they become exhausted, they lose their IL-7 receptor. That seems to be unique to some checkpoint molecules and not others. So each one of them have this nuanced effect. And when you do single cell RNA-seq with these knockouts, you immediately find them out, saying that what are their nuanced effects. But grossly, they look all the same. Okay.
So you talked about the IL-27. The question, would the anti-IL-27 molecule have enhanced anti-tumor effect as it would inhibit all checkpoint?
And in fact, there is a drug company that's exactly what it's doing, is surface oncology, and I always say it's a bad idea. And so they didn't make me the founder of the company, so because I said it's a bad idea. And the simple thing is that the IL-27 is not only inducing inhibitory receptors. I showed you this whole module of also activating receptors. They are part of the module. And we emphasize, we use the IL-27 to get to the inhibitory molecule, but you have all these TNF receptor family members that are part of the module that are induced by IL-27. And then you have IL-21, which is critical for induction of CD8 T cells, expansion of CD8 T cells. So I don't think I will go for IL-27.
Okay, good. So the question from Rakesh Sharda has already been answered on the T cell exhaustion in SARS-CoV-2. Michael Durai wants to know, there are 8 TIM genes in mouse and only 3 in man. But your slide did not show 8, they only showed 7. He was counting those,
Yeah. So we initially defined 7. Then as we got better at it, and we went through this whole genome, and genome got available, the genome sequence got available from the broad, and then we identified there are 8 in mouse. And I apologize, the slide is older, and I haven't updated it. And you're absolutely right.
Yeah,
in the genomic segment, there are 8 in mouse, and there are only 3 in humans. And TIM1, TIM3, and TIM4 are the ones that are actually maintained in the human genome. And
evolution, is it a part of the evolution that?
Yeah, evolutionary, because we have gone through this evolutionarily, and they must be giving some advantage in the mouse. They're still kept, even in the wild mice. We look at the genome of the wild mice. But in case of man, as they evolved, and you can see that the deletions kept happening. And the chimps and then the new world monkeys and humans have only three of them.
So that will be part of the evolutionary advantage of the humans, that they have 1, 3, and 4. I mean,
Yeah, I think there's some sort of inverse evolution that happened, the reverse, that they just actually deleted these. They may not be necessary.
There is a question from Amitabh Sengupta. Does TCF7 regulate TIM3 expression?
Yeah, we showed you the data that there is some sort of regulation, and you'll have to wait and see, as I do not know the real answer. If I still told you anything, it may be wrong tomorrow. So I in the next FIMSA meeting, or when I come to India, hopefully we'll have solved it. We do have this huge screen that we have done on the regulators of both TIM3 and TCF7. And one of these days, I was telling Rafi, I think I met him a few months ago, so I want to show him the data, but we never got time to I never got time to show him the data.
And he also wants to know that therapy with discitabine normally induces ADL1 expression in AML cells. Do you see
It,
that in your
yeah,
trial?
in fact, what it does, what he's correct. It induces PD-L1 expression. It also induces TIM3 and expression. And these are the cells. It doesn't induce, it's kind of interesting. And it goes to something very important in tumors. And we think some of the tumor cells have utilized these checkpoint molecules to protect themselves and make these tumor niches. And it gives them advantage of growth. It comes up, TIM3 comes up on also tumor stem cells in AML. It gives them advantage so that they stick together. And we have a project ongoing on that. How does it give an advantage in the tumor niches? And then I think it is co-opted by tumor to protect themselves and gives them growth advantage. And these cells actually grow faster as well. And this cytokine is killing everybody else. And these are the cells that are actually growing up. Those are the cells you are seeing. And that's why you think it may be inducing, we're doing a toxic profile, but also it gives you advantage and protects you. And that's why I think when you give anti-TIM3 antibody, you're doing two things. You probably, even though our antibody, sabotuzumab, is not an lytic antibody, it is inducing cell death by activating inflammasome in those myeloid cells, tumor myeloid cells. But it also induces anti-tumor immunity. So you have to think of these molecules, even though we think them as immune molecules, they have been co-opted by tumor cells. And the AML cells co-opt them, and we have some data on TIGIT and lung cancer tumor cells, where the stem cells in lung tumors also co-opt TIGIT as a molecule to protect them and promote tumor niches. So it's going to be interesting biology that's going to come up.
Thank you. I think you have already answered the question on IL-27, but Amita from Lucknow is still asking, in autoimmune diseases, IL-27 levels have been reported to be low. So do you think by giving extraneous IL-27 would help?
So it depends on who do you bring out. Do you bring out Jekyll or you bring out Hyde? Because it has got these both faces. It has got this Jekyll and Hyde face. And the IL-27 has been given experimentally in animal models, and with the effects showing that it inhibits EAE, it inhibits psoriasis, but it has not been tested in humans. And I was, in fact, on some of these big pharma are making these agonists of IL-27 receptor. Let's see what happens in clinical trials. And I really worry that depends on who comes out. And I always say, does the Jekyll come out or the Hyde comes out?
So Vijay, although you showed so many activating and inhibitory,
I think in India it will be, does a Ram come out or Ravan come out? Or okay, so go ahead.
Dr. Khan, he says, "Ye dil mange more." How can we identify or screen more regulators?
Okay, so I went through that quickly, and and the question is, do we want more? And we have, if you go to our Nature paper, we have identified something like 53 or 54 molecules in the module.
Yeah. Mm-hmm.
Okay, and we have a paper, we are actually did the screen, and we have identifying these, I told you, this network, what we built in, and we are targeting the network to find how does IL-27 induce all of these 53 of these molecules that look inhibitory molecules, and what are the different nodes and subnodes that you can target, intermediaries. So there are lots of them, and if you want to go, and to your delight, you can go to the paper and you can see all of them.
I will take up just last two, and these could be answered either by you or Rafi. Actually, I think both. I don't know how. What factors should we keep in mind while designing combination of ICIs to minimize adverse reactions and events. This was something that was asked also last time too.
I will ask Rafi to address it because I have been talking for a whole hour. Rafi, you're on mute.
Yeah.
This one is
Yeah,
for you.
I think that's a very important question, Narendra, in terms of the balance between efficacy and
Toxicity.
and toxicity and safety. I mean, this is it's a very. I think there are no easy answers here, and it will depend on the condition of the patient, um, especially when you're dealing with cancer patients. There is a much higher tolerance for toxicity and side effects when it comes to cancer patients who are very ill. So I think this is a topic of great discussion that clinicians have, scientists have, ethicists have. But I think there's no easy answer. And the goal, of course, is always to minimize the toxicity. But sometimes when you do that, you lose also the efficacy.
Yeah, it's
So I
it's a
think
part
this
of,
is
it's
a-
part of the same. It's a part of the same game because
Yeah, I mean, I'll
tox-
just give you one example, which is I didn't go into this, but this data I showed you with the IL-2, we used the wild type IL-2. Uh, we didn't use any of the genetically engineered mutated IL-2s. Uh, and the rationale behind the mutated IL-2s is to reduce toxicity because you then don't have binding to the CD25, the alpha chain. The other reason for that is because you don't activate Tregs. But the bigger concern with IL-2, with wild type IL-2 really is toxicity. Uh, but but the best effect you get on the T cells is with the wild type, is engaging CD25, because that is the pathway for effector differentiation, not the beta and the gamma chain. It's really the alpha chain, which is essential for giving you this. So this is a area that is of requires great thought and debate for many things. I just gave you the example of IL-2, where currently the accepted wisdom is don't use IL-2, which will bind to the alpha chain. That's the current wisdom in the field. But whether it's the right wisdom or not remains to be seen.
Okay, I think we must stop. Although, I mean, the last one, what is the impact of FTY 720 on central memory cells and whether this reduces TH1 like responses? Again,
Go ahead, Vijay.
Rafi,
Vijay,
go ahead.
I think the short answer is we don't know. I mean, the FTY 720 will, of course, prevent, kind of block circulation. And I don't think that many studies have been done. I think probably would not be a good thing, certainly to get an effective response, that's the worst possible thing. But there might be situations where that could be beneficial. But I don't think it's been explored in humans yet.
Okay, thank you so much. I think we have to stop somewhere now, although there are a few more, but I'm sorry, the time is up and I can see that the organizer has already shown up. It's a signal to me to sort of finish. I want to really thank you on behalf of the whole audience here, on behalf of the Indian Immunology Society, the both of you for giving us such wonderful talk. We really enjoyed it. It was a feast for us on Saturday evening for us. Thank you so much.
Thank you. Our pleasure.
Rafi,
Yeah.
thank you for setting the stage. And I,
No,
and I really appreciated it. I
no.
told Sunil, I said, Sunil, let Rafi go first because he will do the heavy lifting.
No.
Actually, it was a great combination of two of you because you complemented each other very nicely. It was great to have both of you in the same session and chaired by Dr. Mehra. So thank you very much for joining, and thank you, Rafi, for agreeing to speak. And Vijay always is very supportive. So whenever I ask him, he comes out very nice. I wish I could invite you to Chandigarh for all these talks, but let's see for a better time when we can invite you here in Delhi, in Chandigarh. Thank you very much.
Thank you for the invitation. Rafi, I got to connect with you.
Yes, yeah. Email me. We'll.
Vijay, I mean, Rafi remembers Goa, so we have to organize something again.
Yeah, I have the money, but I don't have the time. So I haven't given a blank check, but I don't have the time. So let's do it. Let's do it. Let's get over this thing.
Yeah, it'd be good to have a in-person course. Yeah, would be a lot of.
Wonderful.
We will bring you up in Goa.
Yeah.
Okay, thank you very much.
Thank you.
Take care.
Everyone. Bye-bye.
Bye-bye.
Sunil Arora and Amit Avasthi. Amit is
Amit and Sunil.
and his boss is doing wonderfully well.
Bye-bye.
I thank you, Vijay and Rafi. I mean, great talk, and it is just mesmerizing, back to back, two great talks. So thank you very, very much.
You're welcome. You're welcome.
Bye, everyone. Bye-bye.
Bye-bye.
Namaskar.
Take care. Bye-bye.
So,
All right, guys. Now we are back with this closing ceremony, and I would ask Professor Mehra to say something before I close down.
well, closing ceremony is the best thing to do, I really enjoyed. I think it was a great thing that you people did by organizing this course, and I'm very grateful that FIMSA came forward to sort of give this support, necessary stimulus to do it, and you guys have done a great job. And I think the sessions have been very well organized. The topics have been chosen very well, and moreover, the speakers were all very good. I really enjoyed yesterday the session on COVID-19 with Shiv Pillai, and that has reconnected me to Shiv. Shiv has been writing to me, and I've been really enjoying interacting with him. Similarly, I think all these sessions have really gone through well. What we have learned from COVID-19 is that we could organize such kind of things on and off. I mean, you don't have to really worry about finding money for travel and all that. The other thing was that everybody comes on time. Everybody comes on time. This is something that we had not seen in India, and this is something that I see also in the online meetings I do. Everybody comes on time. The meeting starts at 12. It starts at 12. It has to finish at 1. It finishes at 1. I think this is the gain from the virus infection there. But I think I've seen a few more conferences like this being done. We are going to have our own ASHI conference next week. It's going to be all online. And my feeling is that even if COVID goes away, this will become the norm. Such kind of conference will become the norm, because you don't have to really worry for those banners and ये लगाना, झंडा इधर लगाना, उधर लगाना, खाना ढूंढो, ये ढूंढो, सब खत्म. अपना खाना खाओ, अपना चाय पियो, यहाँ आके सिर्फ साइंस सुनो. I think that this has been very, very good. So, Sunil, to you and to Amit, I must appreciate the hard work that has gone behind in choosing the right kind of speakers, moderators, and things of that sort. The whole thing has gone on very well. I feel that the quality of science has been very, very high, and that was the goal. And you guys have achieved the goal. I was only wondering that the numbers should go up even more. Maybe next time when we do, this could be open to others as well. ठीक है, इंडियन इम्यूनोलॉजी सोसाइटी के मेंबर्स आते हैं, कुछ नहीं आते, लेकिन जब इतने बढ़िया स्पीकर ला रहे हैं, it could be open to others also.
It was open to all. Actually, it was by registration only, but open to all. We did not restrict it to members only.
Yes.
It was open to all.
Yeah, but I mean, the ones who have really been the ones who really wanted to.
It's always like that,
Dr. Arora, Monideepa from Durgapur is there.
Hi.
Like, hi, hello. I heartily want to thank you, Dr. Amit and Dr. Mehra also for this nice arrangement. And I am privileged to be part of this one. Hope soon we will be meeting in person, and in webinars also we will be meeting Dr. Gijarilal and everybody. Okay, congratulations. Nice meeting and very enchanting and informative discussions. So you took a lot of pain. You directly contacted us. So it is a huge job you have done, a commendable job you have done.
Thank you.
So,
Thank you. It was
thank
very nice of you that you joined,
Yeah,
and
yeah.
it was a great lecture on blockade. Thank you.
Thank you.
Anybody else from
Bye-bye.
okay, thank you. From the
Bye.
panelists, if somebody wants to say something,
Yeah,
Dr. Sarad Dalai.
thank you, Dr. Arora, Amit. What I experienced, the online is more interacting than offline.
Yeah.
We can see the speaker, we can see their slides, we can really ask questions and get the answers. So,
So totally hassle-free. No
yes,
need to travel, no need for arrangement of tickets and coming in the
it's lot
airport.
more, it's
Yeah.
lot more effective so far learning is concerned. This is what I find. So maybe COVID in a way blessing in disguise.
Right.
Yeah, there are other challenges, but we handled that.
Yeah.
You need some very
So,
strong technical backup. So that's
yeah.
okay. That's all.
And particularly for international speakers, when they find difficult to travel, this is a good way to interact.
Thank you. Thank you, Dr.
Thank
Sarad.
you.
Thank you very much.
I have to go somewhere. Thank you so much.
Okay. Bye. Bye.
Dr. Girdari, you want to say something?
No, I think it was a wonderful program, very nicely done, and I think attendance was also good. But we can think of it in future. Registration is one thing. I don't know how much fund we can arrange, or if we have alternate source to arrange fund. But it should be open also on the social media website or something else.
Yeah.
So
we would have tried.
Can be more.
No, Girdari, actually, I kept registration just to have the more serious ones to join.
Yeah.
Otherwise, there is always a problem.
It is also a very important point. Yeah.
Okay. Thank you very much, all. Dr.
Okay.
Dilafroz.
Bye.
Everybody was so supportive. So actually, I want to thank a couple of people in my team.
Yes, sir. Dilafroz here.
Yeah, hi Dilafroz. How are you?
Hi. So that was such a wonderful course. We really thoroughly enjoyed 3 days of brainstorming sessions. I think it couldn't have been better. So congratulations to you, to Dr. Amit and your whole team. I think a lot of technical stuff must have gone into it, and you rocked it.
Yes.
Great show.
Thank you. I want to thank my colleagues, Dr. Anupam Mittal, Dr. Gurleen, Aryan. They have done a great job. They have helped me, but I did not need very big team. We three of us, we managed so many things here. So it was a great, work by my team. Thank you very much. And I thank all the speakers. That was very nice because I coordinated with them and they were responding very nicely. All the moderators, they did a great job. And my technical team, they did a great job. And thank you very much. Amit, where are you? You say something, buddy.
He disappeared suddenly. Where are you, Amit?
Amit. Hello?
Everyone. Hello?
Amit, say something.
Hello.
Dr. Arora, I want to thank Dr. Anupam personally because he personally called me at the day of my speech, because I sent the PPT, whether it is going okay from here or not, if some problem is facing. So for the safe side, I mailed. Suddenly he called and solved everything. So I want to personally
our job
thank
to support you guys.
Yeah.
That's why I am thankful.
Everybody is so sincere, and in this situation also everything is so well managed. You have done a commendable job, sir.
I think if you have some kind of feedback from the students, those who are registered, they enjoyed or they wanted something else to be added on.
So I've got so many suggestions in the suggestion box. We'll take care of that.
Correct. Yeah.
I'm gonna these all sessions are recorded. They'll be available on Immunology IAS website for a few weeks. So people can see them now live. Today's whole program was on YouTube also, open to all. Today, I just opened it to everyone. And we had it on YouTube also.
Oh, so we can actually look to see the link actually. Anyway.
Yeah, maybe next time if I had funds, I will go ahead free of cost also. This time, because I didn't want to have some money from FIMSA because of FCRA regulations and all. So we thought we'll collect some money which will support my technical, inputs and all, because I had to take license, make a website, and it was interactive website. So all that had to be paid for. So that's why some very token amount of registration was kept. Otherwise, it was all right. We could do it without it.
But Dr. Arora, you have done wonderful job.
Yeah, I think it was.
Oh, thank you. What happened to your power?
I think
I'm just
going back home.
I'm just driving and talking to you.
Oh, wow. Good, good, good. I was missing you. Thank you very much.
You always miss me. I don't know why.
Hey, because
may I ask you a question? You always miss me, but you never invite me to Chandigarh.
Let me tell you.
He knows the reason.
Let me tell you. So listen यार, सुनो मेरी बात. मैं ये कह रहा था कि मेरा तुझसे है पहले का नाता, कोई यूं ही नहीं दिल डुबाता कोई.
अरे ना दारू.
That's nice.
Wow.
So it's got to end with this.
Yes.
It's got to end with this typical note of Dr. Arora.
Yeah. All right. It was
Wonderful
great
friendship
meeting.
they have. They share a wonderful bond. Great.
Yeah.
Yes.
I do. I
Bye-bye. Thank you. Good night.
I value relationships, and I value all of you, your friendship. Thank you very much for all the support, and God bless you all.
And open a bottle and have cheers from my side.
I wish I could offer it.
Amit, family में बैठे हैं.
I wish.
फैमिली में बैठे हैं, अभी फैमिली टाइम है. तुम ये बोतल क्या बोल रहे हो?
यार कोई
I mean
बात
bottle.
नहीं.
I'm saying Coca-Cola.
No, that's okay. I mean,
we all understand what you are saying.
We have to celebrate, and it's all right.
We all understand what you are saying, Amit.
I wish I could. I wish I could offer you all that. If I can have a physical meeting, and definitely I'll do that in future.
We must. We look forward to that. Hope this
Definitely.
Okay.
All right.
Thank you very much. God bless you all.
Weekend.
Bye-bye.
Namaskar.
Bye-bye. Bye-bye.